Okay, last part 6. We're going to continue kind of just going through the cells, identifying these organelles. I'm going to end with incredibly brief talk about protein synthesis. The book is going to go into it in much greater detail. All right, so next we have the mitochondria. It has a double membrane, an outer and an inner membrane. This membrane is a phospholipid bilayer. The function, please, let's not do the powerhouse of the cell. All right, so it's the site of aerobic cellular respiration. Right, that's its function. It produces ATP, site of cellular respiration. Cellular respiration is the process by which ATP is produced. The mitochondria contain its own DNA, and it divides whenever it wants. And I'm not gonna test you on this, but in medical situations, it's not unheard of to access a patient's mitochondrial DNA, because that mitochondrial DNA is an exact copy of your mom's. You get your mitochondria from your mom, and that's in 99.9% of the cases. This is a very rare phenomenon called parental leakage, but for the most part, all of your mitochondria is an exact copy of your mom's. All right, and there's the mitochondria. Yeah, I'm not gonna. All right. Peroxisomes, membranous sacs, and what they do is they detoxify harmful substances, not drugs, right? Because that was the liver. Basically, what they'll do is they'll neutralize free radicals. Free radicals are chemicals that have an unpaired electron, so they are very unstable. They have a lot of energy, and they'll go flying around, smashing into things and damaging them, like DNA. And anytime you damage DNA and it has to be repaired, you're increasing the likelihood that the repair will be faulty, and hence you'll have a mutation. A lot of your antioxidants, what they do is they will be able to bind to these free radicals and neutralize them. All right? Great, cats are awake. All right, cytoskeleton. This is going to be the skeleton of the cell. It's basically a whole bunch of rods that run throughout the cytoplasm, and it really consists of 3 different components: microtubules and 2 others that we're really not going to talk about. All right, really what I want you to understand about the cytoskeleton, one of its functions. Let's say here's our cell, here's the rough ER, and here's the Golgi. How does that vesicle know? How does that vesicle know to get from the rough ER to the Golgi? Well, there are going to be these cytoskeletal elements that serve like a road, and then these vesicles will just walk along that road, and that's how they can go from one spot to another. So, besides providing the support for the cell, it also allows a pathway for intracellular traffic. Okay. And again, we're really not going to be talking about the microfilaments or the intermediate filaments. Our focus is just going to be on these microtubules. Don't worry about that. They help determine the overall shape of the cell. Here's the cell membrane, and then I made mention of this earlier in one of I think the first part, maybe the second. So because this membrane is a phospholipid bilayer, these cytoskeletal elements, in this case these microtubules, provide a scaffolding to give support to this membrane. Remember how I was saying that we have these proteins, and some of the function is to attach to the extracellular matrix and attach to the cytoskeleton. And that's just to help stabilize that cell. And it also is involved in the distribution of the organelles. That's going back to what I was saying with here's the rough ER, here's the Golgi, and then there's a road between the 2. These microtubules can also be involved in the formation of centrioles, and it is 9 triplets of microtubules. I don't care about that. And what they do is they organize the mitotic spindle during mitosis. Super quick. You don't have to know exactly what I'm talking about. I just want you to understand what this mitotic spindle is. If here's this cell, and what's going to happen during mitosis is that we're going to have these microtubules, and they're going to attach. And then what's going to happen is that this half will pull this way, this half will pull this way. So then it will look like this. And that's what happens during division. All of these microtubules here, some that will attach to a chromatid, some that don't, all of them are called the mitotic spindle. So we're going to have this structure right here that's organizing it. And those are the centrioles, and a centrosome is just a pair of centrioles. And we see that here. And here are, I'm not going to test you on 9 triplets, but here are these microtubules. It's just tubulin monomers. They polymerize, and they'll form this long tube, these microtubules. Okay? And here are the paracentrioles, and we call that a centrosome. Next up is the cilia. These cilia are cytoplasmic extensions or cellular extensions, and we see them on the surface of certain cells. In unicellular organisms, such as a paramecium, that's how they move. And in us, we see them in the trachea. And what they do is they help move substances in one direction across cell surfaces. And they too are made up of microtubules. In higher biology classes, you would go over how they operate with those dynon arms. We're not. And so they can, you're just gonna have to trust me, they can contract and bend, and that movement keeps mucus flowing, and that's why we see them in the trachea, commonly known as the windpipe. All right, protein synthesis. There is the central dogma, and that central dogma states the flow of information inside the cell. And what I want you to know is it is a 2-step process. First, we have DNA that will go through a process called transcription. So DNA, here's this DNA molecule. A section of this will be transcribed into RNA, and then that RNA will be translated into a protein. So the central dogma is DNA to RNA to protein, and then I just identified DNA to RNA is called transcription, and that occurs in the nucleus. RNA will then get translated to a protein, and that occurs in the cytoplasm. So, and on this next one, I was about to draw it, but then I realized I have a diagram of it. Okay, so a section of this DNA, a gene, will get transcribed into RNA. It goes through this thing called RNA processing that we're not going to cover. RNA will then leave the nucleus through nuclear pores, where it will be translated to make a protein. And if we're talking about protein synthesis, we know ribosomes have to be involved. And that is it for the cell.